are moving on to chapter 23, the protists. So, we have so far discussed two of the domains of life, the bacteria and the archaea. That means we are on to our final group of living organisms, the eukaryotes. To talk about the eukaryotes, let's talk about the origin of eukaryotes. So, we think about 2.1 billion years ago, this thing happened, where there was a larger cell, represented by the red cell here, came upon a smaller cell, represented by the blue cell. And the larger cell said, I'm hungry. When it's hungry, it would do like it would normally do, this process we refer to as phagocytosis, where the larger cell would wrap its membrane around the smaller cell and then engulf that smaller cell so that that smaller cell was inside this food vacuole, basically inside the bigger cell. But here's the thing. We think that for some reason, this larger cell did not complete the phagocytosis and digest the smaller cell. That it kept the smaller cell inside the bigger cell. Alright, so maybe it went along for a while like that with this smaller cell living inside the bigger cell, and then maybe it ran across another cell. And notice this other cell is green. Green here indicating that it's able to do photosynthesis. And so the bigger cell then said, hey, let me eat that one. But once again, does not digest it, but keeps this smaller cell living inside the larger cell. What we're suggesting here, and maybe this diagram from the textbook is, I don't know, a little more technical, that the organism that only engulfed the first cell becomes a heterotroph, an organism that eats other organisms for food. But then later, when a photosynthetic organism is engulfed, that larger organism can gain that photosynthetic ability and become an autotroph, able to make its own food through the process of photosynthesis. Now, this idea sounds kind of crazy, right? The idea that there are organisms living inside of other organism cells. We do have some evidence in support of this endosymbiont theory, this living inside of another thing. One piece of evidence is that if we look at a couple of our organelles, specifically the mitochondria and the chloroplast, that they have a double membrane. Right? They have a membrane from the original cell that was engulfed, and then the membrane that the larger cell wrapped around it. So a two-membrane organelle. Secondly, if we take a look at that inside membrane, we find that inside membrane is pretty similar to a prokaryotic membrane. Additionally, these organelles go through a replication process that is very similar to prokaryotic replication, to binary fission. In fact, if we look inside of these organelles, we find that they have their own single circular DNA molecule, separate from the larger cell's DNA. They also have their own ribosomes. Not only do they have their own ribosomes and their own DNA, they can actually express that DNA independently of the host cell. And if we take a look at those ribosomes, we find that they are very similar to prokaryotic ribosomes and not as similar to eukaryotic ribosomes. All of these pieces of evidence are pretty strongly in support for the idea that these two organelles are the result of this endosymbiosis, this engulfing of a smaller organism by a larger organism, but that larger organism then retaining it inside and then using it, keeping it as its own little tools for that larger organism to use. And then every time that the larger cell replicates, those smaller cells inside replicate also. Alright, so let's talk about this endosymbiosis. Mitochondria This is this really important organelle that helps organisms to harvest as much energy as possible from their food. We think that the mitochondria derived from an alpha proteobacterium. I remember we talked about that during the prokaryotic section. Chloroplasts, we think, are derived from cyanobacteria. We're actually going to generalize this term a little bit. A chloroplast is a specific thing. We're going to talk generally about plastids. So we're going to say any photosynthetic organelles we're going to call plastids. And so these really important organelles, these organelles that are essential for energy use in living organisms, in eukaryotic living organisms, we are suggesting are cells living inside of other cells. Now, even perhaps crazier, we also think that it's possible then for secondary endosymbiosis to take place, where one eukaryote would engulf another eukaryote. All right, so let's imagine we've got that cell with the plastid and a mitochondria inside of it. And what we're suggesting is, well, maybe another eukaryotic cell comes along, and that other eukaryotic cell says, hey, look at that. Let me engulf that. Let me take that in. But it does so, it doesn't digest it. And now we have, right, a cell living inside of a cell, living inside of a cell. Now, again, this idea also seems kind of crazy, but we have some pieces of evidence. One piece of evidence, something called a nucleomorph. A nucleomorph is a leftover vestigial nucleus. So if we looked inside this red cell, we would find this nucleus that may not be used anymore, but there would be these leftover remnants of that nucleus inside of that red cell. Right? This larger cell would also have its own nucleus, but that red cell would also have a nucleus. It might not be used, might be vestigial, but still be there. We would also find that there are even more membranes. Right? Instead of just two membranes around an organelle, we might have this larger cell has retained this photosynthetic ability, and so now we have a membrane, another membrane, another membrane, another membrane, up to 4 membranes around that single organelle. So, secondary endosymbiosis. Now, as long as we're talking about it, I'll just mention that it's also possible to have tertiary endosymbiosis. So if another larger cell came along and engulfed this one, so then you had a cell living inside of a cell, living inside of a cell, living inside of a cell, and then quaternary endosymbiosis. So if there's endosymbiosis, it would be that next level. And that this sort of thing has happened in the evolutionary history of eukaryotes multiple times. So endosymbiosis is going to be really important for the evolutionary history of eukaryotes. Now, I will also mention, when I first started talking about this, when I first started teaching about 10 years ago, we referred to it as the endosymbiont hypothesis. But this new textbook calls it the endosymbiont theory. This is an idea that has been tested repeatedly. The evidence seems to be in pretty strong support of it and has become pretty widely accepted amongst biologists that the origin of some of these organelles, plastids and mitochondria, are the result of endosymbiosis events, giving the cell that engulfed them then this ability that it didn't have before to either, through mitochondria, harvest energy more efficiently from the food that it eats, or in the case of plastids, to do photosynthesis. All right, well, let's talk specifically then about protists. So protists are this interesting group of eukaryotes. We are going to use the term protist here, but we are going to realize that protist is not a monophyletic term. All right, so we're going to use it every time we say protist. We're going to kind of put quotes around it. We're going to know what it means, but we're going to also understand that it does not include a common ancestor and all the descendants of that common ancestor. All right, it is not monophyletic. We are going to define protist as all eukaryotes except plants, fungi, and animals. All right, so you might recognize this, not monophyletic, because we're defining this by excluding things. We're saying if it's a eukaryote, but it doesn't fall into the group of being a plant, a fungus, or an animal, we're going to call it a protist. All right, so just keep that definition. This is what protists are. They're everything that's eukaryotic, but is not a plant, a fungus, or an animal. Protists are pretty diverse in a lot of ways, including their body plans. Most protists are single-celled. So I think it's like 80–85% are single-celled. However, there are some that are colonial. So the picture on the right here, this is a type of protist called Spirogyra. You can see these lines here. Those are dividing cells up. There are also some that are multicellular. Now, what's the difference between colonial and multicellular? Well, in a colonial organism, we're saying a bunch of single cells that are able to survive independently happen to live together in a group. Right, so this cell of Spirogyra could live by itself, could do everything a living organism needs to do, independently of this cell and independently of this cell, and all the rest of the cells that might be in this long strand of the Spirogyra. A multicellular organism, on the other hand, we're going to have cells that specialize on things. That some cells do something, they give up the ability to do something else, and those cells then end up are needing each other to carry out all the functions of life. So you, as a multicellular organism, have some cells that are focused on, let's say, being skin cells. They can't do what your lung cells do, right? There's that specialization to function. And so some protists have specialization, are true multicellular organisms. There's also some protists that are multinucleate. Multinucleate means basically they're kind of like one really big cell with lots and lots of nuclei, but they're not divided up into individual cells, right? There's this big continuous cytoplasm with lots of nuclei floating around in it, but no divisions between those nuclei. So, kind of weird. We're going to find that for a lot of the things we talk about with protists. Kind of weird. Now, I'll also mention that there are some pretty blurry lines separating these. There are some things that don't really fall into these groups, so just know that if there's some weird way to do something, there's probably some protist that does it. The different life stages of a single protist may differ. Part might be single-celled, part might be multicellular, those sorts of things. So just keep in mind that these are our main categories, but protists are weird. Let's talk about how protists feed. So those that are able to do photosynthesis are photoautotrophs, right? They have plastids. There are some that are not photosynthetic, so are heterotrophs. So here's that phagocytosis that we talked about during endosymbiosis, but this is complete phagocytosis where they actually digest the food particle. So heterotrophs have to eat food, usually other living things, to get their energy and nutrients. And then there are mixotrophs, and mixotrophs are able to do both. They're photosynthetic and they're able to consume food. Now, it's important to realize that we often think about these things, specifically being a photoautotroph versus being a heterotroph, as being sort of a defining characteristic for a type of living organism. For example, if you wanted to divide the plants and the animals, right, you would say, well, plants do photosynthesis and animals eat stuff. It's important to realize, however, that we think, in the evolutionary history of eukaryotes, that being a photoautotroph, a heterotroph, or a mixotroph has arisen independently multiple times. So let me show you what I mean by that. So I'm going to let's imagine that we have a phylogeny here. And so here's our common ancestor. These lines aren't looking all that great. Here are some lineages coming off of that. Here are some lineages coming off of that. And then here's some lineages. And then here's the lineages. I'll tell you, the phylogenies you guys drew for your homework looks a lot better than the ones I'm drawing now. And then here's some lineages coming off of that, and then we can maybe do another one here, and then let's see, that, and then go like this, and then we'll And then maybe we'll do one more here. Okay, so let's imagine that we had this very poorly drawn phylogeny. And we started looking around at the ability to do photosynthesis, which I'll put here, will represent the letter P. So photosynthesis, and then maybe we would find photosynthesis here, and then maybe we would find this species over here does photosynthesis. And then maybe this species here is a heterotroph, and maybe this one's a heterotroph, and maybe this one's a heterotroph, and we'll put a heterotroph here, and then a heterotroph here, and then, so that leaves us with, here's our mixotroph, so maybe there's a mixotroph, and there's a mixotroph, and let's see, that makes this one a mixotroph, and mixotroph, and mixotroph. All right, I think I got all of our species here. Okay, so can I guess I probably shouldn't zoom in. So let's look at this area of our phylogeny here. What we would normally think of is that if you're an organism that does photosynthesis, probably your closest relatives also do photosynthesis. But what we see is that in the evolutionary history of the protists, that ability to do photosynthesis has probably arisen multiple times. Right? And so the closest relative of this heterotroph, of this organism that eats food, is a photosynthesizer. And the closest relative of this heterotroph is a mixotroph. And the closest relative of this photosynthesizer here is this lineage that has both a hexatroph, or a heterotroph, and a mixotroph. So my point is that independently, being a heterotroph, a mixotroph, or a photosynthesizer has arisen multiple times, meaning that sometimes the closest relative of an organism gets its energy in a completely different way. All right, so it's different than what we would normally think of when we divide things into groups, because we normally think of like animals versus plants, where the closest relatives of one animal is going to be another animal that eats other things. But that's not the way protists do it. Protists are just weird, all right? Just accept this at the beginning. Protists are weird. Most protists are able to move. So we have three examples here of protists that are able to move. I'll just mention, since we just talked about it, we have two heterotrophs here and a mixotroph in our examples. Two on the left are heterotrophs, and one on the right is a mixotroph. So these three organisms are all able to move. The one on the far left is going to move through these structures that we call cilia. The one on the far right, we're going to move through the use of a flagellum. Let me just mention that cilia and flagella are structurally identical to each other. Flagella tend to be long, cilia tend to be short. Flagella tend to be just one or a few present. Cilia, there tend to be many cilia present. Flagella are oriented parallel to the direction of movement, right? So you see where this flagellum is. The euglena is going to move this way, right? It's the, mistakes. The flagellum is going to push the euglena this way, so that same direction of orientation as the flagellum. On the other hand, cilia are used kind of like paddles on a rowboat, right? Maybe those are oars. So kind of like a paddle of a canoe, you stick that out to the side and push that way. So perpendicular to the direction of movement. That these cilia on the side are going to be used to move the paramecium, right, left or right. But aside from that, structurally, a cilia and a paramecium, a cilia and a flagellum, as long as we're talking about eukaryotes, they're structurally identical. All right, that leaves one more way for a protist to move, and that's with our amoeba here in the middle. And amoeba use a pseudopod. And a pseudopod is basically it's like a fake foot. Pseudo means false, pod means foot. And so they have this foot, but we're going to call it false because they're able to change it. They're able to put their foot out and pull their foot back. That it's something that's malleable, that's changeable, that's not permanent. And so they could use that to wrap around their prey. They can use it for movement. It's a fake foot that a lot of protists are going to have. And we're going to generally refer to protists that have pseudopods as amoeba. There's also the genus amoeba you can see on the diagram there. All right, well, let's talk about the reproduction of protists for a little bit. And like everything else that protists do, their reproduction is pretty diverse. Some protist reproduction is entirely asexual. So asexual reproduction, they just use mitosis to make copies. All right, so just if you haven't taken BIO330 or just to review, mitosis is just a copying mechanism. It's very similar to what we saw with binary fission. If you start out with an organism that has a single set of chromosomes, that is to say, is haploid, in mitosis, that haploid organism is going to, well, let me find my arrows here. That haploid organism is going to make copies, and though it's just like a copy machine, right? Just like you have a copier that you put a piece of paper in. What do you get from that piece of paper? Well, you get multiple copies of that same piece of paper. So, I know there are arrows here somewhere. Just hold on, bear with me, keep bearing, just a little bit longer. Here we go, I'm about to find them, I'm sure. Is this them? Oh, here we go. Alright, so, in mitosis, if you start out with a haploid cell, you produce more haploid cells. If you start out with a diploid cell, what do you produce? Well, more diploid cells. If you start out with a 247 haploid, that is to say, if you have a cell that happens to have 240 sets of chromosomes, what are you going to get? You're going to get cells produced that are 247 haploid. All right, so mitosis is just a copy mechanism. Some protists reproduce sexually. In sexual reproduction, we don't have just copying, but we have changes that occur. One of the reasons is that sexual life cycles have meiosis. Now, unlike mitosis, meiosis is a process where we start out with a diploid cell and produce haploid cells from it. All right, so meiosis reduces the number of sets of chromosomes. Start out with 2 sets of chromosomes, produce cells that each have 1 set of chromosomes. As soon as you see meiosis, you can know diploid to haploid. We also have in a sexual life cycle fertilization. In fertilization, we take 2 haploid cells and produce a diploid cell. All right, so meiosis and fertilization kind of balance each other. So if you're in a life cycle, if you have meiosis, you have to have fertilization because you have to restore that number of chromosomes during the course of that life cycle. So what that means is that for any life cycle that you're going to look at, you're going to be able to identify whether it's asexual reproduction or sexual reproduction. And let me just mention, there are lots of protists and other organisms that have both sexual and asexual reproduction in the same life cycle. Or if it's sexual reproduction only. And then you can look at it and see whether the organism spends most of its time in diploid state or most of the time in its haploid state. If it alternates between the two, there's a variety of ways that living organisms and protists in general will have those processes of mitosis, meiosis, and fertilization take place during their life cycle. You have the opportunity to work through some of those to kind of see how all that functions and get to interpret some, especially during the labs. And so make sure that you remember those details, though. Mitosis, copying mechanism. Whatever you start with is what you end up with. Meiosis takes diploid cells, makes haploid cells. Fertilization takes haploid cells, makes diploid. Now, I will also mention, because protists are so weird and so difficult, there are other strategies, there are other things that protists do that don't really fit into what we've just talked about. We'll talk about some of them. There's some real weird ones. Protists really like to be difficult. Okay, let's talk about how the protists are related to each other. Now, not that long ago, maybe around the time I was in college, getting to be the end of it probably, there was a kingdom protista. That has since been completely abandoned. There's been various reasons for that. One is that some protists were found to be fungi, plants, or animals. So there were things that we had within the protists that we realized should be actually in the fungi, the plants, or the animals. There were some things within fungi, plants, or animals that we thought they should be, turns out they should be protists. And so we've now abandoned the kingdom protista, and instead we have a level of classification now known as the supergroup. There are 6 supergroups, and the supergroup level is a level above the kingdom. Right? So we talked about this before. We talked about there's the level of domain, and then within domain there are multiple kingdoms. Well, we are now saying that within domain there are multiple supergroups. And within those supergroups then, there are multiple kingdoms. We're going to take a look at the phylogeny. Let me just tell you right now that the phylogeny we're going to use is tentative. It's hypothetical. You know what? I'm just going to go ahead and do it. You don't normally get this in your college classes. I'm just going to say, what the phylogeny we're going to look at is wrong. We have a lot left to learn about how these organisms are related to each other. This is what we're going to use for this class. If you take the class again in 10 years, it'll probably be something different. This is our best understanding right now based on the evidence we have, but we know it's not completely right. We know there are going to be changes that take place. So no more kingdom protista. We're going to divide things up into these supergroups, but exactly what goes into which supergroup, how many supergroups there are, all that is up for debate and is not going to get settled anytime soon. Now, before we look at the actual phylogeny, I also want to encourage and make clear that protists are a relatively, for most people, a relatively obscure group of living organisms. You're probably not going to be familiar with a lot of the terms that we're going to use here. What that means is, in order to remember it, you're going to need to spend some time with it. You're going to need to spend some time making sense of what the groups are, what the names are, even if it's just to become familiar with the names, right? Because I don't know about you, but I don't go around my everyday life talking about, hey, look at that cool chromalveolata, right? I don't say that. So it's important that you spend some time with it to make sense of it so you can kind of get things oriented and just to remember the names of the different things. Let me show you what I mean. So here is the phylogeny we're going to use. So we got our 6 different supergroups here. And probably none of the 6 names are already familiar to you. It's important to spend some time with these so that they make a little more sense. Also, I want to point out, as we start looking at our phylogeny here, that we have this polytomy, right? So we have multiple lineages coming off from this common ancestor. And for a lot of those lineages, we have some dashed lines. Dashed lines mean we're not real sure about that relationship. You might notice that we have those dashed lines on almost all of our phylogenies here. So, almost all of our lineages. So, again, it's going to change. The other thing that I want to point out as we look at our protist phylogeny is that embedded within it are the plants, the fungi, and the animals. And so we're going to talk about plants, fungi, and animals in their own separate chapters. But mainly the reason we do that is because traditionally we've considered those to be separate and special, but you can see they're firmly embedded within these phylogenies, that they are part of the, quote, Protus phylogeny, even though historically we kind of considered them to be separate and different. So, as we're working through, we're going to talk about them in their own separate chapters. We'll talk about them a little bit as we talk about the supergroups to which they belong, but know that they are here, and they are very much part of these groups we'll be discussing. If we wanted to be real technical, if we wanted to make the kingdom Protista monophyletic, we would have to have plants, fungi, and animals added into it, because that would be the common ancestor in all the descendants.